Hello everyone. Today topic is physical base of heredity. So physical base of heredity is nothing but chromosome theory of inheritance. In the year 1900, Mendel paper was rediscovered independently by Corens, DeVries, and Testmark. This paper of Mendel outlined the essential features of the behavior of genes during inheritance. In the same year, the second edition of The Cell in the Development and Inheritance by Wilson was published. This book brilliantly summarized and elaborated the various aspects of chromosomes discovered till that time. It was generally supposed that genes were located on chromosomes, but a critical assessment of the supposition was lacking. Let's see what is the introduction about the chromosome theory of inheritance. So, the idea of chromosome base of segregation and independent assignment was put forth by Sutton in 1902. This hypothesis was elaborated on more fully and was critically evaluated in his 1903 paper. This paper marks with coming together of genes of Mendel and as well established cell organelle, the chromosomes. The synthesis gave us the science of cytogenetics. The hypothesis that genes are located in chromosomes are designated as Sutton-Boveri hypothesis, or more commonly as chromosome theory of inheritance. As indicated above, Boveri is given equal credit for this theory, and he has independently reached a similar conclusion in the same year. According to this theory, genes are the units of heredity and are found in the chromosomes. The chromosome theory of inheritance came into existence long after Mendelian genetics, as we discussed before. They could not believe the existence of such discrete factors such as genes could be segregated without mixing, as this does not support their idea of the constant changes leading to evolution. So here the photographs of Sutton and Boveri in the where they were presented for chromosome theory of inheritance. Next, Reese coincidence Stretchmark discovered chromosomes are exist inside the nucleus. So Walton, Sutton, and Thielman discovered observed that behavior of chromosome when the cells were divided. So there is idea of chromosome segregation combined with the Mendelian principles gave us the theory. So the work was further carried out by Morgan, who used Drosophila to show how sexual reproduction gave rise to variations. So this segregation of chromosome during the segregation of chromosome during the anaphase of cell division. So the two workers were Sutton and Boveri found that close similarity between the transmission of hereditary characters and behavior of chromosome while passing from one generation which means passing from one generation to next generation. So the segregation the chromosome during the anaphase of cell division, which means that in the cell division the chromosomes were passed, they were represented. So gametes serve as bridges between the two successive generations, which means male and female gametes plays an equal role in the contributing hereditary components of further generation. Only the nucleus of sperm combines with ovum. Thus, the hereditary information is contained in the nucleus only. So, the chromatin in the nucleus associated with cell division is the form of chromosomes. Next, any type of deletion or addition in the chromosome can cause structural and functional changes in the living beings. Determination of sex in the most of the animals and plants is affected by specific chromosomes. These chromosomes are called sex chromosomes. Next, there are some features is there for chromosome theory of inheritance. So let us discuss about that. The first feature is that both chromosomes as well as genes occurs in pairs in the somatic or diploid cells. So they will occur only in the pairs, which means they will not occur in single chromosome, single like. Next next feature is a gamete contains only one chromosome of a type and only one of the 12 have a character. And the third one is the paired condition of both chromosomes as well as Mendelian factors is restored during fertilization. Next, transferred from one generation to next, as in the case of genes, which is Mendelian factors. The number of chromosomes is fixed in each living organism. These are found as homologous pairs in the diploid cells. One chromosome from father and other contributed by the mother constitute the homologous chromosomes. So before cell division, each chromosome as a whole and the alleles of the genes get replicated and are separated during mitotic division. Meiosis takes place during gamete formation. So homologous chromosomes from synapses during phase 1 stage, which on later course get separated and transferred to daughter cells. Each gamete or haploid cells has only one allele of each gene present in the chromosome. A characteristic diploid number is again established by the union of the two haploid gametes. Both chromosomes and the alleles, which are Mendelian factors, behave Mendel according to Mendel laws. So some evidences are there for the chromosome theory of inheritance. Before the evidences, let's see what will be the chromosome theory of inheritance was considered with Mendel laws, which was supported by following the followings. Let's see. During meiosis, the homologous chromosomes pair migrate as discrete structures that are independent of further other chromosome pairs, which was discussed before. Each parent synthesizes gamete that contains only half of the chromosomal complement. The sorting of the chromosomes from each homologous pair into pre-gametes appears to in random. The gametic chromosomes combine during fertilization to produce offspring with the same chromosome number as the parents. Every, Even though male and female gametes that are sperm and egg differ in the size and morphology, so they have the same number of chromosomes, suggesting equal genetic contributions from each parents. Let's discuss with first evidence, which is parallelism, which is parallels in the behavior of genes and chromosomes. So each somatic cell contains two copies of each gene. Similarly, each somatic cell has two copies of homologous of each chromosome. So each gamete contains only one copy of a gene or another. The phenomenon that ensures the termed as segregation. Similarly, each gamete receives only one of the two homologous of chrome. So in 1901, McClellan discovered a series of X chromosomes and grasshopper and postulated that these chromosomes was involved in the sex determination. So this was the first demonstration that the character in this case sex, which was associated with a specific chromosome. So this evidence presented thus is quite indirect and relies on parallels in the behavior of genes and chromosomes. Next is sex linkage. Next, next evidence sex linkage. So in 1910, Morgan presented more direct evidence supporting the chromosome theory of heredity. He reported the peculiar inheritance pattern of white eye, white eye in Drosophila, which one of the unusual reported one of the unusual features of this inheritance was follows. So when white-eyed females were mated with, which were in cross B, white-eyed females were mated with red-eyed males. All the F1 males has red eyes, but uh, all the F1 females have red eyes, but all the F1 males were white-eyed. So the pattern of transmission of white eye gene was identical with that of X chromosomes of Drosophila. This promoted Morgan to postulate that the gene W was located in the X chromosome. So this was the first demonstration of an association between a specific gene and a specific chromosome. As in the cross A, they were crossed with a red red-eyed female with a white-eyed male. So all the half of the females in F1 are red, and half of the males are red. So all the F1s are red. So this in the F2 they are showing that 1:1 ratio of red and white. Next, non-distinction of X chromosomes. So let's see what a non-distinction of X chromosome. Uh, Proper distinction separates the two homologous chromosomes during cell division. When there is failure of two homologous chromosomes or sister chromatids to separate during cell division, it is called as non-distinction. So let's see the definition in that. Non-disjunction is a failure of chromosome pairs to separate during meiosis stage 1 or meiosis stage 2, specifically in the anaphase. So here the diagram, here the diagram discovered in the crosses between white-eyed female Drosophila and red-eyed male Drosophila. All the F1 generation should have white-eyed male and red-eyed females. So in 1913, Kelvin Bridges found that there was a rare expect rare exceptions to the results. So he found that white-eyed daughters and red-eyed sons among the F1 generation. Next is uh, attached X chromosome. So in 1913, in 1922, L.V. Morgan discovered a female fly which regularly produced the unusual transmission of sex linkage gene, which are yellow body color. So she concluded that the two X chromosomes of this fly were attached with each other. Cytological analysis of the progeny confirmed this expectation. This further supported the conclusion that the sex-linked genes of Drosophila are located in X chromosome. Next is linkage and chromosome maps. In 1910, Morgan described the phenomenon of linkage and recombination, which is crossing over between two sexual linked genes in Drosophila. A little later, he described linkage between some more sexual linked genes in Drosophila. So, and proposed that the strength of linkage between any two genes depends on the distance between them in the chromosome. In other words, the frequency of recombination between two genes depends on the distance between them in the chromosome. This idea was soon extended to who was proposed the preparation of chromosome maps based on the frequencies of recombination between linked genes. So, extensive linkage maps have been prepared in several organisms like Drosophila, maize, barley, Neospora, yeast, peach, etc. In each in each such case, the number of different linkage groups in the same as the number of chromosomes in the haploid set, not a haploid set. So, not a single case in the known way to differ. This is exactly what could be expected if genes were located in chromosomes. In 1931, Stern in Drosophila and the McClintock in Maize demonstrated that recombination between linked genes indeed involved in the exchange of chromatin material between homologous chromosomes. This was a rather direct demonstration that genes are located in chromosomes. In fact, translocation is used for a handy tool in the assigning linkage groups to different arms of different individual chromosomes. Since structural changes in chromosomes are always accompanied by corresponding changes in the linkage groups, genes must be located in chromosomes only. So, linkage maps of bacteria and viruses are demonstrated to be circular. This led to the expectation that both bacterial and viral chromosomes could also be circular. This isolation of intent bacteria and viral chromosomes have confirmed this explanation. Next is bar locus in the Drosophila. In all the Drosophila flies having bar which means the X chromosomes, at least one of the X chromosome in females has two 16A regions lying side by side, which is tandem repeat. The correlation is a perfect one that it is found in all the individuals having bar eyes. The presence of three 16A segments in a single X chromosome produces the ultra bar phenotype. Thus, a genetic effect was localized to a distinctly recognizable small segment of the chromosomes. Next one is the numerical changes in the chromosomes, which means that haploid or diploiding. Then, in addition to structural changes in the chromosomes, numerical changes also furnish evidence for the favor of the chromosome theory of heredity. In the most cases, a loss is that is monosomy or a gain trisomy of a single chromosome produces marked morphological and physiological abnormalities. In a monosomic form of in a monosomic for a specific chromosome, genes located in that chromosome should be present in a single copy. As a result, in monosomics, such genes showed a marked deviation from the normal inheritance pattern. The same in the case of trisomic plants because they carry three copies of controlled constant genes in the place of the normal two. So this property of monosomics and trisomics is exploited in assigning the different linkage groups to individual chromosomes of the haploid set. In a large number of diploid plants, autotetraploids, autotriploids, etc., have been experimentally induced. Some plants are known natural autoploids. Example: alfalfa, potato, coffee, sweet potato, banana, etc. So all such plants could be expected to contain more than two copies of genes. So three copies in triploids, four in tetraploids, and so on. Consequently, they are expected to show some complex inheritance patterns than diploids. So for even monogenic traits. In fact, natural tetraploids show a complex pattern of inheritance, which can be explained only on the basis of presence of four copies of the concerned genes. Next is Y-linked inheritance. So some genes, example SRY genes in humans are located in the Y chromosome. Such genes are transmitted from father to son and are absent in females. Characters produced by Y-linked genes are known as Hollandric genes, and since they were confined to males only. Next is genetic material. It is accepted beyond any reasonable doubt that DNA is a genetic material in eukaryotes and in prokaryotes, except in some viruses. It therefore follows that the organelles containing DNA must contain genes and vice versa. By far, the vast majority of DNA occurs in the chromosomes of eukaryotes. It is therefore self-evident that genes must be an integral part of eukaryotic chromosomes. Next is in situ hybridization. Gene expression occurs as follows In the first case, RNA polymerase produces RNA copies, which means mRNA of one strand of the gene. Second, in the mRNA direct synthesis of a specific protein, and in third case, this protein participates in cellular activities to produce the phenotypic specified by a gene. So radioactively labeled DNA copies of a DNA different mRNA molecules have been shown to base pair, which is hybridized with the DNA specific DNA different but specific locations of chromosomes. This technique is called in situ hybridization. This demonstration beyond any doubt that genes are present in the chromosomes. So the last evidence is the transgenic organisms. The mRNA produced by a gene can be used to obtain copies of the concerned genes as for concerned genes, like the enzymes reverse transcriptase can be used to produce a DNA copy, which is complementary DNA of the mRNA using latter as a template. Next, alternatively, the genomic DNA, which is DNA present in chromosomes of the organisms, may be isolated, and the genomic DNA segment complementary to the mRNA may be obtained. Examples like in the genes obtained by the either technique have been transferred into a different organisms where they are expressed. So copies of genes obtained by other approaches produce identical proteins. So the foregoing considerations clearly demonstrate that there is a little doubt that genes are located in the chromosomes. This conclusion is universally accepted as one of the fundamental facts of genetics and cytogenetics. Let's see uh, the genes obtained by either technique have been transferred onto different organisms. So that clearly shows that genes are located in the chromosomes only. So here the topic ended with the chromosome theory of inheritance. Thank you.